I'm Mallory Embry, and I'm the Chief Science Officer and co-founder of Native Microbials.
I'm Jordan Embry, and I'm the Microbiology Manager at Native Microbials. And as you can see behind us, we're at our lab, and today we're going to be showing you a tour of the lab to see the whole process of product creation from beginning to end.
This is our sequencer. Next-gen sequencing really enabled our technology. It actually lets us go into rumen content and actually sequence the microbes there so we understand what microbes are present, what their abundance is, and how that changes over time. This technology is really special because it enables us to get gigabases of data in just a matter of days. This is our fax machine. It stands for Fluorescent Activated Cell Sorter.
What we're using this machine for right now is to determine whether or not our microbes are releasing from the encapsulate. So in the studies that we've designed for this, we'll take one of those encapsulated microbes, we'll put it into a buffer solution simulating rumen fluid, and then over time, periodically take samples and run it through the fax. What the fax will tell us is how many bacteria is in that liquid that we're flowing through it. It does that by using lasers to shoot into a stream of liquid that's being brought through the machine, and the laser will hit a bacteria, it'll register as an event, and it'll plot it onto the screen here. So by determining how much liquid is being flowed through the machine and how many events are populating, we can count the number of bacteria from that original sample and determine how much bacteria is releasing from our encapsulate.
Back in 2015 and 2016, we had an entire team of anaerobic microbiologists who spent all of their time culturing microbes from the rumen. And all of this effort was to actually try and isolate out the product microbes because they don't exist in culture collections today. Jordan has some plates that he streaked out.
Yeah, so what I did was I took raw rumen fluid that we took from a cow, serial diluted it, and then plated it onto a variety of different agar medias that have different carbohydrate sources, kind of common things that cows are eating because that's the microbes that we're trying to isolate. So we'll plate the rumen fluid on that. The organisms that can grow in that media will grow. And then after about a day or three, we'll get these colonies growing, and from those colonies, we'll individually pick them, put them into liquid media, grow them up, and then sequence them to determine what they actually are. Because in the state that they're in right now, they're just white and gray circles on an agar plate, and it's difficult to discern who they actually are, what genus and species of bacteria these are.
After we isolate the microbes that we want to turn into product strains, we give them off to Rob, who is part of our fermentation team.
So here at fermentation, we'll take these novel microbes that have never been investigated, never been grown, and try to figure out how do we make them economically and large quantities and stably so that we can take them to final product.
So after fermentation, we have to preserve our microbes. Megan, what does this machine do?
So after fermentation, we concentrate our cells down into packed cells, and then we mix them with preservation solution that is comprised of sugars, and that basically replaces the water during the lyophilization cycle. These are our lyophilizers, and they freeze dry our cells and take them from liquid to solid. And after that, they look like this. They're a powdered form so that they can remain stable on farm conditions where it's really hot and humid.
We have some final products ready for you guys to visualize how do they look like. So this is we call it MPBV, preservation by vaporization. This is one of our strengths. So this is after the fermentation, after lyophilization, and how those powders look like. And then after encapsulation, they look actually Like you might not feel a big difference, but actually their particle size is much larger than the previous one. So they have been protected and encapsulated pretty well. So this is our pre-blend, like intermediate material. So this type of prototype will be blended with other components in our product, and then they're gonna survive much longer than this original version.
Figuring out the stability of our products is super important, so we have an entire team just running experiments that basically validate the stability of our products under a variety of conditions. So you can see here we have a number of different incubators set at temperatures all the way up to 50 Celsius to kind of represent the worst case scenario. And when we run these experiments, we actually package them in smaller containers that are basically similar to the real packaging, right? So we get a true ground truth measurement of what the stability of the microbes are over time.
So our quality department determines the concentration of microbes within our product. Jeremiah, what are you working on here?
So right now, I'm working to enumerate our DY21 strain. So this is our spiral plater. It does a lot of our hard work for us. So our initial concentration in our bags are too concentrated, for lack of better words, to enumerate. So we have to dilute it down to a smaller dilution in order to get countable colonies on our plate. So this is what the spiral plater is going to be doing. So right now I'm going to be diluting to 10 to the minus 3. So I'll plate that. And then the spiral plater will basically add buffer to these 3 cups and transfer our starting sample, which I already previously made, and do series of serial dilutions all the way up to 10 to the minus 3. And then it'll grab 50 microliters from our last sample and plate it onto our plate. And basically I'll rinse and repeat and do that until all our lots are done.
And then for the big picture, this is really just determining the concentration of microbes within our product. So when we make a minimum label claim of a specific concentration of microbes, we are 100% sure that that's what we're delivering.
After we do spiral plating that we just showed you from the quality department, we let the plates incubate for a couple days, and then they come to Jackie, who then counts the colonies.
So basically, we take one of the plates and we put it into the camera right here. And on the screen, it'll show us the colonies. And after that, we'll hit count, and it'll automatically count colonies. It'll count the first 20 colonies in the outer rings. I will either select or deselect any of them that are colonies or not. And this is how we make sure that the colonies are alive.
So in order to have consistent fermentations at our contract manufacturing organizations, we have to have consistent stocks of all of our microbes. So our fermentation team produces in-house all of their vials that they'll be using to grow our microbes up at scale. So each time they make these vials, they make approximately 300 cell bank vials that they'll ship off, ferment at large scale, and produce to make our product. So for every customer that we have, we run a TMR audit to show that they're mixing appropriately. Our product is a 5-gram inclusion for each animal, and the testing that we do is called a microtracer assay. We mix 50 grams of TMR into 2,000 pounds of feed, and we'll take 10 samples from that and try to pull out as many of those magnetic particles as we have. What Rachel's doing here is she's weighing out a feed sample. We usually weigh out about 200 grams. From that 200 grams, she'll put it into a mixing bucket. She'll add water to that feed and mix with a magnet in the feed to try to extract as many microtracer particles as she can. Once the mixing's complete, she'll pull the magnetic tracers off the magnet onto a filter paper. evenly distribute it onto a different piece of filter paper, and then activate the dye that's conjugated to that magnetic tracer with a 7% sodium carbonate solution. And once you add heat to that mixture, then the microtracer will release its dye, and we can count that blue circle that appears to determine whether or not the feed is being appropriately mixed. And that will tell us whether or not each animal, each cow on the farm is getting the right amount of product in their feed.
That was a tour of our facilities here in San Diego, where we do all of our microbiome research. If you have any other questions, please feel free to reach out.